Now that I spoke a little bit about this, let me go down to the next step, which is start creating the experiments and the biosamples. So I will scroll down to the experiment section, and you see pretty much everything is the same. You go to a section, and in the section to the right, you have an add records. So I will click here, add records, and start creating my first experiment. So first I will go to the special genomics experiment I want to define. So I will search here spatial genomics and select that term. So you will see for all the metadata terms, I mean for most of them, well, we have sort of drop-down menus that show terms that exist in a control vocabulary. So we avoid as much as possible free text fields. So pretty much every metadata field comes from a well-known ontology whenever possible, or a control vocabulary. So we have control of which terms are being used to tag the data, and whenever possible, we use well-known established ontologies. So in this case, the experiment type is spatial genomics. So I will give here a local identifier. This is something that, so that people know, give some information. If for some reason your organization have a well-defined scheme to do this, then you can use that term. If not, for example, I will create a, I will give it a name, spatial genomics for, hard palette. This is, I use the hard palette for this, okay? So here, if I will click save here, that will define my first experiment. But something that you will see will use and is very handy and allows you to generate entries quickly in our UIs is this feature of cloning the element. So here I will click clone once, and will allow me to generate the next experiment that I want to use, which is a single cell. So again, I go to the experiment type, I select the single cell RNA sequencing experiment type, and here I will change the identifier so that I know what it means. RNA seq, and this one I did use the soft palette. So now I click save, and if I go back to my data set, I will see that there's two new experiments appear. Something I wanted to show you is that, when I went to create the experiment, it created a new tab, and then I can navigate back to the data set either by going back going back to the previous tab. In other parts, you will see that you can navigate back by following a link, and I will show you that later too. So right now, I basically created my two experiments. Now I will go into each of these experiments and define the biosamples associated that are corresponding to that experiment. So let's say that in the spatial genomics experiment, I have two biosamples associated to that. So I will scroll down to the section that says biosamples and click add records and start creating my first biosample. So like if you see, the first two fields are the data set and the experiments that this biosample will correspond to, and those get automatically populated. And then there are two fields. One is the subject ID and the other one is a local identifier. Subject ID is something that we mostly use for humans. So in this case, since it's a mouse, I will just give a local identifier. And again, if in yours lab, and we've seen that a lot, people have well-defined schemes to identify biosamples, then we encourage you to use that scheme, whatever you use in your lab notebook or something like that. If not, you can just add something that is descriptive. For example, this is going to be E12.5, hard palette, mutant, okay? Because I will have, I pretend that I have one mutant and one control for this type of experiment. And then I'm gonna start adding all the metadata that we want to associate to this data. And we have all these drop-down menus that help us do that. So species, the first one, I will select mass musculus because it's a mouse. A specimen type, I think the cryostat slice sample is probably the closest one for this type of experiments. And then, for example, let's say we have gene, probably doesn't make a lot of sense, but let's say that this is a gene that is the gene of interest, because we know whether we have sequencing data, you have a lot of genes that your data contains the data for all the genes, but let's say you have a specific gene that you want to focus on, you can add it here, and that will help people discover this data set. So let's say I will assume these are ACAN mutants. So I will select ACAN, and I don't know if you noticed, but all these terms come from this NCBI ontology. So we have pretty much a whole NCBI terminology in here. So most likely you will be able to find your gene. And then next one is the genotype. Let's say this is a mutant. I'm not 100% sure that this makes biological sense, but I will assume that this is a genotype is ACAN minus. Then we can enter the strain, mutation. If you have all the information, again, we have vocabularies for that. Then I will select the stage, where I say this is a E12.5 mouse, so I will select E12.5. Anatomy, I will select a hard palette, and you see we have all the terms from the Uberon ontology, which is pretty comprehensive. So you can have a lot of choices to select to describe the anatomy of your biosample. Then we have a lot of other fields that some of them mostly are more appropriate for human data. And then, for example, let's say we collected the data today, okay? So that's probably all the information that I can provide for this biosample right now. Like I said, I have two of these, and the other one's pretty much everything's the same, but it's the control. So I will clone this biosample. In the local identifier, I will change mutant to control. And in the genotype, I will change that to wild type. And select wild type. And then I click save, and I go back to my experiment, and I see that the two biosamples for that experiment are now created. Then I can use, for example, this link to navigate back to the data set. Scroll down to the experiment section and do the same with the second experiment for the single cell RNA sequencing. So I dig into the experiment for single cell RNA sequencing, scroll down to the biosample section, and I will start adding my first biosample for single cell. So I will pretend that these are E15.5, soft palette. This is a mutant, and I have two replicates. So I will use the rep one to say that this is the replicate one for that mutant. I will select mass musculus again, specimen. Right now I'm going to use single cell gene ACAN. And we understand that this can be time consuming and tedious to select all this data, but I think we found that once people get familiar with using this, they can add a very large number of biosamples and describe them very quickly. So don't get intimidated. I'm pretty sure once you start, you will see that you will agree with what I'm saying. So I will select the genotype again. Stage is E15.5. Anatomy, we said this was the soft palette. Find it here. Again, another return from Uberom. And I will say that the collection date was today. So for this experiment, I want to define actually 4 biosamples. So what I will do is I will use this clone feature again, select clone, and now I have 4 biosamples, and I only need to go and do the small changes that I need. So for example, the second one is going to be the replicate 2 for the mutant. The third one is going to be the replicate 1 for the control. And the fourth one is going to be the replicate 2 for the control. Probably the only other changes that I need to do are the genotypes of the control. So in the genotype field, for those that are control, I will go again and enter wild type. And I'll do the same here. And again, I will click save, and that defines these 4 new biosamples. Now I navigate back to my experiment. I can navigate back to my data set. Something I want to show you, on the top right, we have this very useful button here that says hide empty section. It's very useful when you're entering data, because if you click it here, it will get rid of everything that is empty and get rid of all that clutter and see what you have done so far. So what we have done so far in this last 15 minutes is basically generating the experiments and the biosamples, and you see that all the biosamples show to which experiment they're associated, and all the experiments show the description of the biosamples, the metadata associated to the biosamples that correspond to those experiments. So the only thing that is left now is basically upload the data files, associate the files to the biosamples, and then add the protocols to the experiments.